Since we're studying evolution, and evolution depends on genetic variation, which in viruses is caused through a mutation, I thought it'd be a good fit to start with taking a bit of a closer look at COVID-19. That's the thing that spread across the planet, shutting down universities, including ours, all kinds of sports organizations, and social gatherings worldwide. So technically, it's the SARS-CoV-2 virus that causes the coronavirus disease 2019, which is also known as COVID-19. The virus is an RNA virus, which means it has ribonucleic acid as its genetic material. So RNA viruses have a really high mutation rate, which makes them really easy to track but difficult to make an effective vaccine. So the errors occur as it gets passed from individual to individual and has an estimated mutation rate of about every second or third transmission. So the mutations happen during the life cycle of the virus, which is shown here in this diagram. So the SARS-CoV-2 virus infects person, and through the replication complex that occurs within the host, mutations can arise, and then the new SARS-CoV-2 gets transmitted to the next individual. So the mutations that occur are random, and they occur as part of this natural viral life cycle. And some mutations are detrimental to the virus, and some are beneficial, but mutations themselves are random. So researchers are tracking the evolution and spread of the virus by sequencing its genome and identifying novel mutations. Although most of the genomes are really similar, there's some cautious interpretation of the patterns that are evolving, and those mutations allow researchers to track its movement, time, and space. So I'm going to pause for a second here while I go to this website, which is tracking the spread of the virus in near real time. So the website that I'm showing here is allowing the public to see what's happening with the genomic research that is going on with COVID-19. So scientists take buccal swabs from infected patients, then they sequence the genome of the virus, and they can track the individual mutations or changes in the virus's genetic code as it spreads from person to person. And these changes have been described as fingerprints that help researchers chart its movements across the globe in near real time. So once they sequence the genome, they construct a phylogenetic tree, which you can see here. And that helps them understand the evolutionary pattern of the mutations and allows researchers to infer the global spread of the virus. So some of the things that are interesting to note here is these early sequences here, and in this you can actually click on the particular sequence, and you can see that this sequence came from Wuhan in China, and a lot of these early sequences are from Wuhan in China, and they're very genetically similar. So those are the early samples that occurred with the virus outbreak. After Wuhan, we see it actually appearing in other places in China. For example, Guangdong province in China is shown here, and then eventually we see a spread to elsewhere in China, and then in February and March you can see areas like Italy and Europe and other places experiencing outbreaks. So one of the interesting things is most of these originate through a genome that's very similar to the original genetic outbreak that occurred in Wuhan province in China. The other interesting thing to note is if we come up here, we can actually see that the North American viruses are indicated in red here, and if we come up, we can actually see some of the Canadian cases that can show the actual divergence and the date that it was established and who is actually publishing these particular viruses to help understand more about the spread of the virus in hopes to develop a vaccine against the virus. So take a look at that. I think it's kind of an interesting and relevant topic for the course that we're taking right now, and I hope you find that kind of informative with respect to mutations in RNA viruses.